Regulatory T cells are critical in autoimmunity, and their therapies are mainly focused on enhancing their suppressive activity. FOXP3 is a key marker for regulatory T cells, and its stable expression is required for regulatory T cell maintenance and immunosuppression. Loss of FOXP3 in regulatory T cells partially causes autoimmune diseases. FOXP3 stability depends on the demethylation of the CpG motif in the FOXP3 locus. Studies have shown that certain inflammatory conditions can induce regulatory T cell instability with diminished FOX3 expression and convert them into pathogenic effector cells. Thus, the identification of new targets crucial to the plasticity and function of regulatory T cells is of importance in the development of therapeutic approaches in autoimmunity. Protein phosphatase 6 belongs to the serine threonine protein phosphatase family. Protein phosphatase 6 specific deletions in regulatory T cells leads to a marked impairment of regulatory T cell induction. A study published in Genes and Diseases examined the effect that conditional phosphatase 6 knockout has in regulatory T cells. Researchers found that conditional phosphatase 6 knockout in regulatory T cells led to a spontaneous autoinflammation, immune cell activation, and diminished levels of FOXP3 in mice T cells. 3 kinds of DNA methyltransferase enzymes are responsible for DNA methylation, one of which is DNMT1. In mice, DNMT1 deficiency in regulatory T cells weakens their suppressive function, suggesting it is crucial to regulatory T cells. The researchers proposed protein phosphatase 6 as a critical positive regulator of FOXP3 that acts by decreasing DNA methylation of the FOXP3 gene enhancer and inhibiting AT signaling, thus maintaining regulatory T cell stability and preventing autoimmune diseases. Mouse interleukin 17A ELISA kit and mouse interferon gamma ELISA kit were used in this study to quantitatively analyze pro-inflammatory cytokines. Anagen is willing to provide hundreds of high-quality antibodies and ELISA kits to support continuous advancements in biomedical research. For more information, please visit Anagen's website at www.anagen.ca.